Isolating a single pustule.
When you take a collection from the field, that collection could actually be a mixture of races. So in order to create an isolate, you need to derive just a single pustule and collect spores from that single pustule in order to get an isolate that can be screened for race analysis. So this pot has been inoculated with stem rest, and you can see that there are different pustules or different spots of infection on these plants. What we like to do is isolate a single pustule, and what you can do to do that is first cut away all of the plant tissue except for a single pustule. Now you can see that we've cut away all of the plant tissue except for a single leaf and a single pustule on that leaf. Now what this single pustule is an infection from a single spore of rust. Now collecting from this single pustule will allow us to get a genetically pure isolate of rust. In order to isolate spores from just a single pustule that you've cut away on a plant, what you can do is place this plant with a single pustule of rust into a dew chamber for 5 hours. Now if you do not expose the plant to light while in the dew chamber and allow the plant to dry after being in the dew chamber for 5 hours, the spores will germinate in the dew, but they will not successfully infect the plant because of the absence of light. If you take your pot with a single pustule after this dew chamber period and place it into one of the isolation cubicles, you can allow the rust to grow for 1 to 3 days. At this point, you can go in and collect spores from the single pustule. And so I'm going to demonstrate how we would collect the spores from just this single pustule using a gelatin capsule.